Okay. Now we'll be quick on these. These are really what? They're not fungi. What are they? Bacteria. Okay. Three genera. We'll discuss very briefly. Here are the important points for you to know. That Actinomyces, that's the genus Actinomyces, is an anaerobe, and it produces granules. Okay? Nocardia are partially acid-fast. The other two are aerobic. And they are closely related to the mycobacteria now. That's all been shown very well biochemically and analysis of the organisms. Now, as you know, do you have mycobacteria already? You had that, didn't you? They're acid-fast organisms. These nocardia are partially acid-fast. What that means is the organism will stain unevenly with the acid-fast stain. So a knowledgeable TB person or mycologist can tell the difference by looking at them. They're usually gram-positive rods, so they look like a bacterium microscopically, but they're kind of long rods and they're branching, and that's why they were thought to be fungi. Actinomycosis is a chronic suppurative disease. In cows, it's called lumpy jaw because it produces a big lump in the jaw. Frequently, a patient has a tooth pulled. The Actinomyces organisms are endogenous. You get a tooth pulled, and of course the cavity will be anaerobic. So the organism will grow there and an infection occurs. They also occur on the trunk of the body, but that's more rare. They're usually in the cervical area. The one organ, there are a number, but Actinomyces israeli is the primary organism. Endogenous in man and animals. Now, this is a 6-year-old boy from Augusta, Georgia, who had lumpy jaw. Now, the thing that had to be done is take the pus and examine it. This is a Petri plate, and that's pus in there. You can see the red blood, and the yellow are the granules. We call them sulfur granules because right in the beginning they look yellow like sulfur looks, and they were thought to be sulfur. You see all those shining? That's it. So you can take some pus and make the diagnosis right in your office or bedside, whatever, just by looking at the pus. And if you got the symptoms, that's why you got to have a prepared mind. This is a granule, microscopic, crushed. And see, a lot of these are filamentous structures coming out, and all these are polymorphs, pus, suppurative reaction, like you would think looking at them. And it's treated with penicillin. What was I just telling you about antibiotics fungi antibiotics don't work? Why is penicillin treated? Because they're not fungi. No serological tests. Incision. You can't just treat with antibiotics. You got to drain all that pus. You got a lot of pus, whatever the disease is. You got to drain it out first. And this boy recovered very well. Now, nocardia will cause 3 types of infection: spontaneous, subcutaneous infections, pulmonary infections, often confused with tuberculosis, and brain abscesses. It has this predilection for these areas. It's in soil, and it's thought to be endogenous because in the TB lab, you isolate a lot of these from a patient that does not have nocardiosis. You get a lot of nocardia because nocardia will go through the horrible test of that you have to do, the procedure to isolate tuberculosis. It survives, and it's partially acid-fast like TB. So it's thought to be endogenous, and they occur worldwide. Remember endemics, that's why I'm going to start telling you about where these things occur. For some reason, one species, Nocardia asteroides, is mostly pulmonary disease, and the other species and abscesses. That's not a test question, but Just try to keep it in the back of your mind. Pulmonary disease looks like TB, and it looks like a lot of the mycotic diseases we're going to talk about. And subcutaneous abscesses. These drain and form another abscess and drain, and they often get secondarily infected with other bacteria that cause chronic diseases. What you send to the lab, the sputum, pus, or biopsy tissue, depending on how the disease is presenting itself. Now, here are some. They look like mycelium hyphae fragments, but they're really bacteria, and no serological test. Sulfamethoxazole trimethoprim is the drug of choice. Again, an antimicrobial, not really an antibiotic. It's a chemical antimicrobial. And the third genus is Streptomyces. There are many species here, and we just discussed them as a group. They are definitely in the soil. When you dig in the soil and you get that odor, you say, this is a soil odor. That odor are the actinomycetes in general, particularly Streptomyces. That's the odor. When you grow it in a plate, you uncover the plate, you get this odor of soil. That's the Streptomyces odor. And Streptomyces is where many of our antibiotics come from. Different organisms of different species of Streptomyces produce many of the antibiotics. The first antibiotic, penicillin, was developed from a fungus, Penicillium. The second one, Waxman isolated in the late 50s, and that was what? Streptomyces, come on, say it. Streptomycin came from Streptomyces. Here's one. This is well healed now, but you can see where the lesions were, all up and down this leg. This is in the healing process. Now, the one thing about Streptomyces is they can invade the bone. This is an X-ray of this patient in Central America where he had all these lesions. Again, they're pretty well healed now. But you see the X-ray, and these are lytic lesions in the bone. See the lytic lesions? So Streptomyces can invade the bone. Again, biopsy material or pus, depending on how the patient presents. They produce granules, and they'll produce all kinds of granules, different species. That's one of the ways you identify them. You look at the granules. They can be yellow, white, black, red, and they can have a smooth wall or be textured in one way or another. So, and this is all pus cells. This is a pyogenic reaction. This is a crushed granule with the GMS stain. You remember the black stain? Gomri methenamine silver. And you see it even stains them as if they were fungi. This crushed granule with a GMS stain. No serological tests. First thing is surgical debridement, sulfa drugs, and amphotericin B.